Good morning, girls. It's okay now.
Yes, Miss. Good
Good morning,
morning.
Miss.
How are you, girls?
Fine, Miss. How
Alhamdulillah,
are
fine.
you?
We are going to start our class, and today we'll solve past paper from 2016. Before studying my class, I want to tell you one thing, that today is, if you check the date, today is 9th April, right? And the school has instructed you through the teacher that you have to settle the matter paying the tuition fee within 10. Otherwise, you are not allowed to enter the class, and tomorrow is the last day. And still, tomorrow is Friday, you can utilize the weekend, no problem. But from Sunday, those who are not paying, you are not allowed to enter the class. So I'm requesting you, please, this is a reminder, talk with your parent to settle this matter. All the students should pay till March. Otherwise, school is not from Sunday, they will not, they're not allowed that student to enter the class. So did you get me? Girls, understood?
Yes, Miss.
Okay, so please inform your parent to settle this that matter so that the school can run smoothly. Now, we are going to start from 2016, 41 October, November 2016. Could you please open October, November 2016, variant 41. October, November 2016, variant 41. And question number 3. Syed Ishraq, what happened? This is the third time I'm allowing her to enter. Anyway, now, so have you got the piece, girls? Are you ready?
Yes, Miss. Yes, Miss.
This is page 9, question number 3.
Miss, can you please repeat it one more time?
October, November 16, variant 41. October, November 16, variant 41, question number 3, page 9.
Is it pepsin is a protease enzyme found in the element?
Yes. So the first question, pepsin is a protease enzyme found in the alimentary canal. Name the product formed from the digestion of the proteins by protease enzyme. When the
Amino acids?
Yes, excellent. When the protease enzyme will break down the protein, it will turn into amino acids. Yes. State the organ in the alimentary canal where pepsin is secreted. Girls, I told you, pepsin secreted. Now, please switch off your mic. So pepsin.
Page number 9. Why can't I? Oh, wait, October, November?
Yes, October, November. You have to listen carefully. And then state the organ in the alimentary canal where pepsin is secreted. That is stomach. You know that, and from the pancreas, that
Miss.
I know if it's making too much sound. I have to mute her.
Miss, what is A1? Like 3A?
Amino acids. Amino acids and then stomach. Just still this 3A you have finished, then I'll go for B. Still you are not in. Okay, shall I start question number B?
Yes, Miss.
Okay. A biologist performed an experiment to find out the optimum pH for activity of pepsin. The enzyme activity was measured in 4 test tubes. Each test tube contained 1 cubic centimeter cube of cooked egg white, which contained protein. This figure 3.1 shows 4 test tubes. Look here. There are 4 test tubes. The biologists measured the time taken for the complete digestion of the cube of cooking egg white. So what they have done, see, in first test tube 1, that first test tube, they have given pH 1, then pH 3, then pH 5, then pH 7. So different pH solution they have given to the test tube. And the cube of cooked egg white they have also given. Now the question is B1. The biologists ensured all the cubes of cooked egg white were exactly the same size. Why? You know, girls, because of surface area. If that pieces, the cooked egg cube was different in size, so enzyme will work in differently, because the more surface area, the more enzyme contact will be there with the exposed area. So that's the reason. So more contact means more pH reaction, and for the faster the result. So if you want to get the fair result, you have to make sure that the surface area are same. That's why they have taken the same size cube. Now I'm dictating the answer. Please write the answer. Because of same surface area. Girls, because of same surface area.
Only because of same surface area.
I didn't finish. I mean,
no. Like this is the starting, right?
Yeah, write the answer. Because of same surface area. Full stop. If surface area is different, if surface area, yeah, that because of surface area, full stop. If surface area is different, it will affect the enzyme activity. It will affect the enzyme activity, and biologists could not find the optimum pH.
Miss, can you repeat? It will affect the...
Okay, sure. If surface area is different, it will affect the enzyme activity, and biologists could not find the optimum pH for pepsin. Done. Now next question.
Miss, could you repeat the last sentence?
Yes. Biologists could not find the optimum pH for pepsin. Now, the next question they're asking, temperature must be controlled in this investigation. And you know that if you're working that to find out that effect of enzyme or pH, temperature also is a factor that is affecting the enzyme activity, right? So describe how temperature could be controlled in this experiment. If you want to control the temperature, how can you maintain this?
Thermostatically controlled.
Yes, by using a thermostatically controlled water bath. Answer, by using thermostatically controlled water bath.
Using
thermostatically controlled water bath. Now we will be moving on next page. Next page, this question.
Miss, can you repeat?
By using thermostatically controlled water bath. I can't understand Saida Ishraq. This is 4th time. I have to admit her. Saida Ishraq, where are you going every time?
Miss, I'm so sorry. It's my internet.
And this is the fourth time, maybe continuously coming and going, coming and going. And sometimes, as I'm busy with the paper, sometimes I've not also noticed that computer board also. So,
Sorry,
okay.
Miss.
The same experiment was performed with trypsin, another protease enzyme, which has been secreted from the pancreas. You know this, right, about the trypsin. And I told you trypsin couldn't work in acidic condition. That's why sodium hydrogen carbonate also secreted from the pancreas to neutralize the acidity, and also bile, which also neutralizes and gives an alkaline medium. So what would be the pH for trypsin?
pH 8.
8, excellent. Alkaline pH. Thank you, pH 8. You can, pH 7 actually neutral, right? So we will not write pH 7. So pH 8 or pH 9 you can say. pH 8 is good. Now, this answer question, you can see total 6 marks, right? So girls, first I will explain and you will not write. Now, I will tell you when you need to write.
Miss,
Yes, Saida.
Miss, I'm sorry to interrupt, but could you repeat B2?
B2.
Yeah, the previous page.
Where are you in the time? B2.
Miss, my internet. I told you I lagged.
Okay. Internet was
So sorry.
disturbing, right? Yeah, sometimes it happens. No problem. B2 answer is by using thermostatically controlled water bath. By using thermostatically controlled water bath. Now, is done. Shall I go? Question number D.
Yes, Miss.
Now, don't write right now. Listen first. Enzymes such as proteases are important in digestion. That's we know for protein digestion. Describe in detail how enzyme function using other digestive enzymes as examples. See, they are asking you enzyme function, and you have to use here all the digestive enzyme. Now, first, what we are going to do here, because the question is 6 marks, we will first give some, that normal characteristics of enzyme. Then we will start how this enzyme start working as that in our elementary canal. Now, listen, enzymes are protein. They are specific and they can be used again and again. You learned this thing in enzyme chapter, right? So enzymes are biological catalysts which speed up by lowering our activation energy and that needed for the reaction. All the enzyme has active site which is specific for the substrate. Because of their complementary shape, the substrate fit together and form enzyme substrate complex. So now I have given, because the first part of the question they are telling, describing detail how enzyme function. So we have given how enzyme function. Now using other digestive enzyme as example. So I will tell you now other digestive enzyme. Digestive enzyme perform chemical digestion and break down large insoluble molecule into small soluble molecules. Now I will give an example. Like amylase enzyme convert starch to glucose, lipase converts fat to fatty acid and glycerol, proteases like pepsin and trypsin break down protein to amino acid, maltase convert maltose to glucose. But high pH and temperature denature the enzyme. Understood? So detail function of the enzyme you have to give for 6 months. Is it clear? Girls, understood the answer? Hello?
Yes.
Yes, miss.
Now you will start writing. Now start writing. Enzymes are protein. Comma, they are specific and can reuse and can reuse again.
Miss, can you repeat?
Enzymes are protein. They are specific and can be reused again.
Miss, can be.
Yes, they can be used again if you need, and if the enzymes are not denatured. Enzymes are protein, they are specific and can use again. Full stop. Then you will write enzymes speed up the reaction. Enzymes speed up the reaction by lowering the energy needed for the reaction. By lowering the energy needed for the reaction. Enzymes speed up the reaction by lowering the energy needed for the reaction. All enzyme has active site. All enzyme has active site for specific substrate.
Means energy needed for
the reaction. All enzyme has active site for specific substrate. For specific substrate. Full stop. Because of their complementary shape. Because of their complementary shape, enzyme and substrate fit together and form. Enzyme substrate complex.
After substrate.
Because of their complementary shape, enzyme and substrate fit, F-I-T, fit together and form, F-O-R-M, and form enzyme substrate complex.
Miss, what's after form?
Form enzyme substrate complex. Done. Full stop. Digestive enzymes perform chemical digestion and break down large excuse me, Safa.
Miss, after chemical digestion?
Digestive enzyme perform chemical digestion and break down large insoluble molecules into small soluble molecules. Large insoluble molecules into soluble molecules. Full stop. Amylase convert starch to glucose. Amylase converts starch to glucose, comma. Lipase converts fat to fatty acids and glycerol.
This amylase convert
Starch to glucose, comma. Lipase converts fats to fatty acids and glycerol.
convert
Lipase converts fats to fatty acids and glycerol. Full stop. Proteases like pepsin and trypsin. Proteases like pepsin and trypsin break down protein. Proteases like pepsin and trypsin break down protein to amino acids. Proteases like pepsin and trypsin break down protein to amino acids. Comma, maltase convert maltose to glucose. Done.
Can you repeat?
Sure. Proteases like pepsin and trypsin break down protein to amino acids. Maltose convert maltase, M-A-L-T-A-S-E. Maltase converts maltose to glucose. Who is this? Suho is back. Can anyone give me the idea?
Safa. Yeah, Miss Safa.
But you should come in proper ID. Otherwise, I'll not be understand who is this. So girls, this is the answer. So last line will be maltase enzyme convert maltose to glucose. So this is the answer. Next page, page number 11. Question number 4. It's done, girls?
Yes, Miss.
Question number 4. Type 1 diabetes, this is not in our syllabus, but they suggest which organ in the body attack. This is not in the syllabus. That antibody function also not in the syllabus. But describe the cause of vitamin D deficiency in human. Look at question number B1. What is the reason that the human have vitamin D deficiency?
Lack of sunlight.
Lack of sunlight. Excellent. Lack of sunlight. And girls, that B2 describe the effect of vitamin D deficiency in human for 2 months. Lack of vitamin D causes rickets, right?
Muscle cramps.
Yes.
Muscle cramps.
So you need to
Stomach
muscle cramp, fatigue. Yes. But the very important point is rickets.
Rickets.
Yes. Lack of vitamin D causes rickets in which bones become soft and deform. Then you can write it also causes muscle cramp, fatigue. Also affect the growth, whatever. But this point you must write. Lack of vitamin D, girls, lack of vitamin D causes rickets.
My second one. Second one.
Yes, B2. Describe the effects of vitamin D deficiency in human. So lack of vitamin D causes rickets, in which the bones become soft and deformed.
Means
Lack of vitamin D, Amira. Where are you?
Means I have a very crazy brother who is studying also in the same room, and he's shouting. That's why you can hear him through the laptop speakers. I mean, sorry, headphones also.
Okay, okay. Lack of vitamin D causes rickets, in which the bones become soft and deformed. It also
soft and
soft and deformed. D-E-F-O-R-M-E-D, deformed. And it also causes muscle cramp that you told, muscle cramp and fatigue.
Miss, will I write that?
Yes, because 2 marks, you need to write 2 points. You explained about the rickets, now you will tell about the muscle cramp and fatigue. That's it. 2 different points it has to be.
And Miss, for B1, it is lack of sunlight, right? Or whatever.
Lack of sunlight, yes.
Miss, after muscular muscle cramp?
That yes, cramp and fatigue, that's it.
Cramp and?
Fatigue. Tiredness. Now, we'll be moving on October number 16, variants 4-2. And I don't know, this question will makes you tired. Next question. Get ready, girls. Question number 1. Don't be scared.
Can you repeat the variants?
Yes.
Oh my God.
See, I told, Sadeshak understood. Oh my God. October number 16, variant 4-2. Question number 1, page 2.
My hand is going to break.
Detail. Describe in detail how enzyme function to digest protein in alimentary canal. And they gave you 6 marks for this. And they gave you almost full page to write. Anyway, no problem. We'll start from the beginning. So milk is sometimes referred. Page 2, question number 1. October, November 16, variant 4-2, question number 1, page 2. Milk is sometimes referred to as a complete food because it contains all the nutrients that our young mammals require. Now, this table 1.1 shows 3 nutrients that are contained in milk. Complete the table by stating 1 role each nutrient in the body of a young mammal. See, girls, if you memorize the notes, the past paper question answer, you can be able to solve. Now, see, this is direct from the notes. Protein, what is the role in the body?
For growth and repair.
Tissues.
Repair, that's it, growth and repair. Lactose. Lactose is
Provides energy.
Provides energy by respiration.
Good. Ari, who is this? Exactly, Marcus Kim answer is telling. Calcium.
Miss, provide.
For strong
Bone and teeth.
Or your strong bone and teeth. Yes, any one you can write according to your wish. Now, this is the big question. Protein digestion begins in the stomach of the human alimentary canal, and it is completed in the small intestine. That's also true. And you know that different proteins, enzymes are also working, different pH that matter also there. So first, listen. You will not write now. You will listen the explanation, then I will tell you to write. So describe the detail that how enzyme function to digest protein in the alimentary canal. See, enzymes, again, we will give that few lines about the enzymes. Enzymes are biological catalysts which speed up the reaction. It also lowered the energy needed for the reaction, right? So in stomach, the gastric juice secrete enzyme pepsin, right? Can you remember, girls? In stomach, the gastric juice secrete in pepsin enzyme. That protein fits into the active site of the pepsin because of their complementary shape and form enzyme substrate complex and break down the protein to polypeptides. I told you in the stomach, protein couldn't break down completely amino acid. It will break down a small molecule from protein to polypeptide. At the end of the reaction, the enzymes are unchanged and can reuse. It carry out the chemical digestion and break down large insoluble molecules into small soluble molecules. In duodenum, what happened? In duodenum, trypsin activated and break down protein to polypeptides and then to amino acid. In ileum, peptidase enzyme break down polypeptide to amino acid. Pepsin work best in acidic condition, so stomach pH is 1 to 3. Trypsin and peptidase can work best in neutral or alkaline condition, so their pH is 7 to 9 and in the duodenum, and they need nearly 37 degrees centigrade temperature for their successful collision. So understood, girls? Actually, both this all is from the notes. Only enzyme little bit function you have to add, which is from the enzyme chapter. Understood? Shall I start?
Yes.
Okay, now it start writing. In stomach, right, in stomach, the gastric juice gastric juice secrete enzyme pepsin. Gastric juice secrete enzyme pepsin. Protein fits into the active site of pepsin because of their complementary shape. And form FORM and form enzyme substrate complex. Which break down protein and form enzyme substrate complex which break down protein to polypeptides.
Miss, breakdown
Amira.
protein. Yes, miss. It wasn't me, miss.
And break down protein to polypeptides.
And form polypeptide or what?
Yeah, and break down protein to polypeptides.
Sorry.
Miss, which breakdown protein?
To polypeptides. At the end of reaction, enzymes are unchanged. Enzymes are unchanged and can reuse. At the end of the reaction, enzymes are unchanged and can reuse. Reuse. Full stop. In duodenum, DUODENUM. In duodenum, trypsin activated. Trypsin activated and breakdown. Girls, I'm coming because this is they're showing less than 1 minute. I'll give you the link again. So from duodenum, I will start again. Thank you. I'm coming back. Okay, good afternoon. girls, that we were doing that October number 16 question number 1, that is a 4-2 variant, right? So I told you that we are going to start from the deuterium function. So are you ready? Shall I start? In deuterium, trypsin activated and break down protein to polypeptides. Hello.
Yes, miss.
In deuterium, trypsin activated. In deuterium, trypsin activated and break down protein to polypeptides.
Maria.
In deuterium, trypsin activated and break down protein to polypeptide, then to amino acids. Then to amino acids. Full stop.
Oosh. Okay.
Then to amino acids, full stop. In ileum, in ileum, peptidase enzyme, P-E-P-T-I-D-A-S-E, peptidase enzyme breaks down polypeptide to amino acids. Polypeptide to amino acids. Full stop. Pepsin work in acidic condition. Pepsin work in acidic condition in stomach, where the pH is 1 to 3.
Is it 1, 2, 3 or
1-3? You can write like that.
Okay.
And trypsin and peptidase enzyme.
Miss, repeat.
Trypsin, T-R-Y-P-S-I-N, trypsin and peptidase, P-E-P-T-I-D-A-S-E, trypsin and peptidase enzyme work best in neutral. Someone having that password problem, maybe, girls, because that's the reason I.
It's only 8A here. Only 8A.
Oh, I didn't give this link 8D.
I
Okay.
think so. Because it's all 8A.
No problem. I will just repeat this question. No problem.
So we sit down.
Okay.
Best in
neutral and alkaline condition. Neutral and alkaline condition in pH 7 to 9. 7 to 9. Don't worry, I'll be coming back for 8A because your testing was late, no problem. 8D, right? I'll coming back to the answer. Let them write. So the in deuterium and ileum. That's it, this question.
pH
So
7-9,
yes,
pH 7-9.
7 to 9 in duodenum and in ileum. Done. Answer is done. So girls, those who are from 8D, that in duodenum, 8A, you just stop for a few minutes. Girls, in duodenum, trypsin activated. Still there for me. In duodenum, trypsin activated and break down protein to polypeptides. In duodenum, trypsin activated and break down protein to polypeptide. Grade 8D.
Yes, Miss, continue.
Okay. So in duodenum, trypsin activated and break down protein to polypeptide and then to amino acids. Full stop. In ileum, peptidase enzyme.
Miss, can you repeat? Protein
Yes,
to
sure.
polypeptide and?
Then to amino acids. Then to amino acids.
In ileum.
Then to amino acids. In ileum, peptidase enzyme breaks down polypeptide. In ileum, peptidase enzyme break down polypeptide to amino acids.
In ileum?
Peptidase. In ileum, peptidase enzyme breaks down polypeptide to amino acids. Full stop. Pepsin work in acidic condition. Pepsin work in acidic condition in stomach. Where the pH is 1 to 3. Trypsin and peptidase work best.
Miss, where the pH?
pH 1 to 3. 1, 2, 3. Full stop. Trypsin and peptidase.
Can you repeat after in acidic condition?
In stomach, where the pH is 1 to 3. Full stop. Trypsin and peptidase work best in Trypsin and peptidase work best in neutral.
Work best in?
Trypsin and peptidase work best in neutral and alkaline condition. In pH 7 to 9. Neutral and alkaline condition. Done, last line. Neutral and alkaline condition in pH 7 to 9, comma, in duodenum, and ileum. Done.
Miss, can you repeat the last line?
Sure. Trypsin and peptidase work best in neutral and acidic condition in pH 7 to 9, comma, in duodenum and ileum. Done. This question has over. Now, I'll be moving on page 3, question number C. Girls, did you hear about lactose intolerance? Do you know that when that small the kids are that couldn't eat the solid food, mostly they are taking that milk, right? The milk protein is lactose. You know this thing. But sometimes what happened, you know, that when the people have grown up, this enzyme, the lactase enzyme, which digests the milk protein, is not produced in their body. So these people, those who do not produce, their body do not produce lactase enzyme, if they have taken milk or that milk product or milk, the food made with the milk, then this will not digest in their body. So they will have severe indigestion, that problem, pain, stomachache, the tummy upset problem will be there. So this kind of problem we call lactose intolerance. And these people, if they're not taking milk or the milk made any food, they'll not have any problem. So this question is that about lactose. Some people are unable to digest lactose or the milk sugar and have a condition known as lactose intolerance. This is, I told you, lactose intolerance is the problem. Some people, the body, their body is unable to produce lactase enzyme, so they couldn't digest the milk protein, which is the lactose. Lactose is milk protein. Now, this diagram shows, yes, dear.
Miss, why does this happen? This lactose intolerance?
You know that why does this happen? Their body is unable to that produce this enzyme, and mostly Asian descent, this type people are suffering this problem. Maybe for climate or any other reason, it stopped working. But when the people were small, the person was small, in that time it was producing, because till 1 year that babies are mostly taking milk only, right? So this is the diagram. Look at this diagram. This is the large intestine and the small intestine. Look, the small intestine from the stomach, and then also they're showing the large intestine. It shows what happened in the intestine of a person who is lactose intolerant if they eat food containing a lot of lactose. Look, so from the stomach, the big the pieces of this lactose is coming, the milk sugar is coming and entering to the large intestine. Bacteria produce hydrogen, methane, and carbon dioxide. Bacteria digest lactose to make it simple sugar. And now what happened? This bacteria
Miss,
and this
are we
lactose,
doing the question with
C? Yes, C. Now this action of the bacteria, those who are trying to break down these big molecules of the lactose, it will build up gas and water in the large intestine, and this will make this tummy upset problem. So this is actually the diagram they have showed. Now the question is, why lactose is not absorbed by the small intestine? Because no lactase enzyme. Girls, don't right now, listen. Because no lactase enzyme to break down lactose into small molecules. Because if it is not a small molecule, it will not be absorbed through the villas, and it will not go to the blood. Through the blood capillary, it has to through the hepatic portal vein, it has to go to the liver, and then it will go to your body cell. But it will not happen because the molecules are large, it is not soluble form, and it cannot pass through the membrane. So girls, and there is no carrier protein for the lactose, girls. So this is the reason. Now you will write the answer. Everyone, please. No lactase enzyme. 2 points only we need to write. No lactase enzyme to break down. No lactase enzyme to break down lactose. No lactase enzyme to break down lactose into smaller molecules.
Miss, one more time.
Sure. No lactase enzyme
Miss, you're doing number 1 or number
no
2?
lactase enzyme to break down lactose into smaller molecules.
smaller molecule.
Because lactose is too large. Because lactose is too large and complex molecule. Because lactose is too large and complex molecule and cannot pass through the cell membrane.
Miss, because lactose is too large
and cannot pass through the cell membrane.
Then where is complex molecules?
Because it's big. It is not simple. After breaking down, it will be formed from complex to simple molecule, right? Simple soluble form. It's complex molecule. So it is large and complex. So it cannot pass. So it cannot pass through the cell membrane. Done. Now, next one more question. That is
Please
actually
repeat the whole answer one more time.
okay, sure. No lactase enzyme to break down lactose into smaller molecules. Because lactose is too large and complex molecules and cannot pass through the cell membrane. Now go page number 4.
Miss.
Yes, miss.
Sorry to interrupt you, but can you tell me question A, like the second one, lactose sugar milk, what is that?
Could you please ask your question again?
Question number 1, A.
Yes,
A,
1A.
lactose milk, the second one. Yeah, 1A.
This is, yes, milk sugar. Lactose milk sugar. The milk also
Provides energy by respiration.
and every time if it is sugar, doesn't matter that milk sugar or that that rice sugar, any sugar is providing you energy only, right? By respiration. But if the lactose is not breaking down in your body, how it will provide the energy?
She was asking for the answer in the
A.
Okay, now I don't know, Sumaria, your sound is very low. I couldn't hear properly. No problem. Next page, go to page number 4.
This is
the last question. Last question for today. I will just that even though class 10:30, thus do you have any class right now? Break, right? So 2-3 minutes extra I will take. Suggest
for this question.
Yes, for this question, because
Yes,
this year I want to
question.
finish. Suggest the dangers to health of severe diarrhea if it is not treated for a long time.
You die.
Yeah. So that 3 points we need to write, right?
Miss?
Yes.
I wrote a different answer for the answer question 1. After writing this question, can you listen to the answer?
Which question 1?
The previous one, the one question.
Question
C1.
1A.
C1.
C1. Okay, just check. No lactase enzyme to break down lactose into smaller molecules. Because lactose is too large and complex molecules and cannot pass through the cell membrane. That's it. Okay, next one. Severe diarrhea causes dehydration.
Oh, yes.
Right? Severe diarrhea causes dehydration. Dehydration in body.
Miss, could you start over, please?
Severe diarrhea causes dehydration in body. And loss of ions.
Miss, causes
dehydration in body and loss of ions and loss of iron decreases the volume of blood.
Decreases the
Volume of blood and blood pressure.
volume.
Yeah, volume of blood, because osmosis problem. Can you remember? Decreases because whenever you are going to lose the ion, then your concentration changes. Decreases the volume of blood and blood pressure.
Miss, I'm sorry, can you repeat after loss of iron?
Decreases the volume of blood and blood pressure. Full stop. It causes slow metabolism. It causes slow metabolism, comma, kidney failure, and brain cell may damage. Done.
Miss, what's after kidney failure?
It causes slow metabolism, kidney failure, and brain cell may damage. So girls, this is the answer. So I will start from 2017 next class. So please, again, I'm reminding you all, please clear your school fees. Otherwise, from Sunday, you are not allowed to enter the class. So sorry for that. I have taken 5 more minutes in this class. I couldn't finish, that's why. So till 16, we have completed the past paper. Okay, girls? So see you next week.
Okay.
And don't forget to memorize the notes and all. Okay. I'll be taking next week there, but if I that next week, most probably Thursday, I'll take a small class test, not so big. Okay.
Okay,
Okay, thank you, girls. Bye. Have a nice weekend.
Miss, to you.
Okay,
You
bye.
also. Bye, miss.
Bye, miss. Bye, miss.